Let's think about this for a second. Deep inside almost every single cell in your body, there is a sprawling, highly coordinated manufacturing center. And it runs continuously, literally day and night, just to keep you alive. We call it the Electron Transport Chain, but honestly, it's more like a breathtaking microscopic factory. So as we dive into this explainer, we're going to see exactly how this cellular machinery extracts energy from your food and transforms it into the literal spark of life.
Just look at that massive number, 32. When you eat something, let's say a piece of bread, your body eventually breaks it all down into glucose. But, crucially, it doesn't just burn up that glucose wildly like some out-of-control campfire. No, through the miraculous process of oxidative phosphorylation, the complete oxidation of just one single glucose molecule yields approximately 32 molecules of ATP. And ATP? That's the fundamental energy currency of life. It powers your heartbeats, your muscle movements, even the very thoughts you're having right now as you listen to me. Getting 32 ATP per glucose is an absolutely incredible return on investment, and it's all thanks to the microscopic factory we're about to explore.
Now, we're all familiar with that classic bean shape of the mitochondria from basic biology, right? But the reality is way more complex. When you actually look inside, the mitochondrion features this intensely folded inner membrane. These intricate folds, they're called cristae, and they aren't just there to look cool. They are structurally designed to maximize surface area. It's what allows the cell to pack tens of thousands of individual molecular assembly lines into just one single organelle. In highly active tissues, like your heart muscle, these cristae are packed incredibly densely just to keep up with the massive energy demands. For decades, figuring out exactly how this factory actually stored its energy was one of the greatest puzzles in all of biochemistry. Enter Peter Mitchell in 1961. He proposed his groundbreaking chemoosmotic theory, and let me tell you, it was a massive paradigm shift. Mitchell proposed that the energy extracted from biological oxidation reactions isn't just stored in some floating chemical intermediate. Instead, it's stored as a physical state of tension, a transmembrane difference in proton concentration. Essentially, the energy is stored as this massive gradient across that folded membrane we just talked about. Today, this stands as one of biology's absolute greatest unifying principles. To really grasp Mitchell's theory, we have to understand the proton-motive force. Simply put, it's the electrochemical gradient created by pumping protons across an impermeable membrane. This creates a massive reservoir of potential energy. Think of it kind of like compressing a giant heavy spring, or pumping millions of gallons of water way up into a high tower. This proton-motive force is the literal, physical energy reservoir that powers human movement, thought, and survival.
Section 1. The electron assembly line.
Okay, so every factory needs raw materials, right? Here, the raw materials are high-energy electrons, which get stripped right from the food we eat. But electrons can't just float around the cell freely. That'd be chaos. They need secure transport. As you digest your food, catabolic pathways strip away those high-energy electrons and load them into water-soluble carriers, primarily NADH and FADH2. You can think of these guys as microscopic delivery trucks. They drive straight to the inner mitochondrial membrane at breakneck speed to drop off their volatile cargo. And here's the precise, highly organized path those electrons take once they're dropped off. It's basically a microscopic bucket brigade. First, NADH donates its electron packages to the massive complex I. From there, a tiny lipid-soluble molecule called coenzyme Q acts kind of like a forklift, shuttling the electrons over to complex 3. Then another shuttle, Cytochrome C, quickly ferries them to complex 4. And finally, molecular oxygen steps in at the very end of the line to accept those electrons, neatly forming water. Now, you might be wondering, why do we pass these electrons along in such a complicated bucket brigade in the first place? Well, as electrons move from carrier to carrier, they are spontaneously flowing downhill to lower and lower energy states. This is an exergonic flow, meaning it releases energy. The factory then captures that released energy to perform an incredibly difficult endergonic task, uphill pumping. It literally uses the energy of the falling electrons to forcefully pump protons against their concentration gradient, moving them from the mitochondrial matrix up into the inner membrane space. So the downward flow directly fuels the upward push.
Section 2. The membrane pumping stations.
A really great way to visualize this entire process is to think of a hydroelectric dam. The river is the continuous flow of electrons toward oxygen. As this river flows, it powers the pumps, which are the giant multi-subunit protein complexes I, 3, and 4 embedded right there in the membrane. These pumps relentlessly push water up into a reservoir, which represents the massive proton gradient building up in the intermembrane space. And finally, all that pent-up water flows back down through a turbine, the ATP synthase, generating our usable energy.
Let's look closer at one of those actual membrane pumps in action, Complex 3. It essentially acts as a massive pair of mechanical doors orchestrating something called the Q cycle. It catches the electron-carrying coenzyme Q from the membrane, physically shifts its own shape, and mechanically ejects protons right across the membrane barrier into the intermembrane reservoir. It handles this complex transition from 2 electron carriers down to 1 electron carriers with absolutely stunning mechanical precision.
But remember, electrons don't just magically jump over empty space between these massive complexes. They have to be carefully ferried. That's where Cytochrome C comes in. It's a small, soluble protein that just slides along the outer surface of the inner membrane. It acts as a highly specialized mobile shuttle, carefully carrying just one single electron at a time away from Complex 3, ensuring it safely reaches its final station. Without this careful ferrying, loose electrons would wreak havoc, causing massive cellular damage and oxidative stress.
And that brings us to the climax of the electron's journey, Complex 4, or cytochrome oxidase. This thing is a behemoth of an enzyme, containing 13 distinct subunits in mammals. It catches the electrons from cytochrome C using these highly sophisticated copper and heme centers. Then it holds on tight to an oxygen molecule, literally the oxygen we breathe in, and patiently transfers 4 electrons to it, step by step. By doing this so carefully, it creates 2 molecules of water perfectly and safely without releasing any dangerous free radicals into the cell. It is a miraculous feat of biological engineering. And of course, it uses this massive energetic reaction to pump 4 more protons outward to the reservoir.
Section 3. The Proton-Motive force in action. When we put it all together into a master schematic, you see the entire factory floor functioning continuously, all at once. Electrons are constantly entering at complexes I and 2, flowing through their mobile carrier Q, moving into complex 3, then riding cytochrome C over to complex 4 where they finally meet oxygen. And while all that horizontal electron flow is happening, protons are continuously, relentlessly, being pumped vertically out of the matrix and into the intermembrane space. We are building an immense difference in chemical concentration and electrical charge. We have officially built our proton-motive force.
Section 4. The ATP turbine.
Now it's time to meet the ATP synthase. As those thousands of protons we pumped up into the reservoir rush back down their gradient, they cause the core of this specific enzyme to physically rotate. I mean, it spins like a literal high-speed mechanical turbine, revolving at thousands of times per minute. This violent spinning changes the shape of the F1 knob that protrudes down into the matrix, causing it to rapidly cycle through 3 distinct states, grabbing ADP, slamming it together with a phosphate to form ATP, and then ejecting the finished product.
But here's a fascinating twist for you. You might think the energy of those falling protons is used to chemically bond the phosphate to the ADP, right? Actually, no. The chemical formation of ATP on the surface of the enzyme requires very little energy at all. The ATP practically forms spontaneously once it's in the pocket. But the enzyme holds on to that newly minted ATP incredibly tightly. So the true job of that massive proton gradient isn't forming the bond, it's providing the violent mechanical force needed to wrench the enzyme open and physically pop the finished ATP off the machine so it can actually go out and power your cell. Mind-blowing, right?
Your entire life is completely dependent on this microscopic perfection happening inside you, right this very second as you listen to this explainer. We've seen just how elegant and tightly coordinated this mitochondrial factory is. But what happens when these pumps become leaky or the turbines start to slow down? Well, the consequences are severe. A decline in this exact process is directly tied to aging, diabetes, and major neurodegenerative diseases. Every single breath you take relies on billions of these microscopic assembly lines running without a hitch. If the proton gradient fails, life stops in seconds, which really makes you wonder what happens when we intentionally or accidentally introduce everyday chemicals that short-circuit this incredibly delicate factory.
Thank you for watching. See you in the next one. Stay tuned.